disorders are inherited from the chromosomes assigned to people. These happen by either abnormalities in chromosome number, having either too few or too many, chromosomal structural rearrangement as well. This can cause huge differences in the offspring by changing many genes or even being fatal to zygotes made. In fact, mistakes are so common that around half of all embryos produced never make it. It's quite possible that a woman can be pregnant and miscarry and never know about it. In order to clinically diagnose chromosomal disorders, a karyotype can be made by extracting and placing these chromosomes in order by size, banding pattern, and centromere position. In humans, chromosome 1 through 22 are called autosomal chromosomes and are placed in order by size, with chromosome pair 1 being the largest and chromosome pair 22 being the smallest. You'll notice the 23rd pair is known as the sex chromosomes, or X and Y chromosomes. These are not autosomal. In this particular karyotype, this person has 2 X chromosomes, making them a biological female. Biological males have an X and a Y chromosome. And again, for those of you watching and planning to misrepresent the information I'm giving, please understand that biological sex in mammals, birds, and insects is determined genetically. However, in other groups of animals, biological sex can be determined by external factors like the temperature of the nest, stress, and even just to balance out sex ratios within a population. And so while chromosomal designation provides an easy explanation for biological sex in humans, it is quite possible that the environment of the uterus during development, as well as external environmental factors, could also play a role in the biological sex and gender expression. Also, some people are actually born with both sets of sexual organs and/or chromosome types. The point is, it's complicated because, well, living things and genetics are complicated.
We also need to get into a few more terms dealing with what's called ploidy, with euploidy first. Euploidy is when an individual has the correct number of chromosomes for their particular species. For example, a human with 46 chromosomes. Aneuploidy is an individual with an error in chromosome number. One could be monosomy, which is the loss of a chromosome, or trisomy, which is the gain of an extra chromosome. This one's a lot more common in the chromosomal disorders seen in humans. For example, Down syndrome, also known as trisomy 21, is the result of a gamete undergoing non-disjunction, which I'll talk about later, resulting in having 2 copies of chromosome 21 instead of 1 copy. So in this particular egg, instead of having 1 copy of chromosome 21, there are 2 copies. And when the sperm, which has 1 copy of each chromosome, unites with that egg, that extra copy of chromosome 21 from the sperm is added to this now zygote, and this individual now has 3 copies of chromosome 21, giving them Down syndrome. Down syndrome causes a short stature, stunted fingers and toes, a broad skull, large tongue, and developmental delays. There is a strong correlation of non-disjunction leading to Down syndrome the older a woman's age is. And that's because as people age, their sex cells are more likely to have mistakes in the chromosomes that are being produced during meiosis. Polyploidy results in individuals with more than the correct set of chromosomes. And this would be like a human having 92 chromosomes, which is double to 46. Polyploidy is extremely rare in animals, but if it does exist, it results in animals being sterile, since meiosis can only produce aneuploid gametes. In plants, polyploidy is actually really common. In fact, natural strawberries that are 2N pale in comparison to the robustness of the strawberries we eat due to the centuries of selective breeding. And that's because these strawberries are octoploid, or 8N, meaning they have 8 sets of chromosomes. So sex chromosome aneuploidy can occur in many different forms. So I'm going to give 3 examples in humans. First is Klinefelter syndrome, which develops XXY males. In other words, it's a male that has 2 X's and a Y chromosome. And this will result in males with taller body build, narrow shoulders, smaller testes, and infertility. Females with Turner syndrome have monosomy. In other words, their karyotype only consists of 1 X chromosome, so they don't have a second X chromosome. And they're usually sterile and have underdeveloped sex characteristics. Now, while chromosomal abnormalities can be caused by chromosome number, sometimes all 46 chromosomes are there, but pieces are missing or are duplicated. And this can result in physical or mental disabilities. One example of this is the cat's cry syndrome, in which a portion of chromosome 5 is completely gone. It's called cat's cry syndrome because of the really high-pitched cry made by infants, but it does create anatomical abnormalities as the child develops. So how do these chromosome disorders arise? They all come from a process called non-disjunction, which is the failure of homologous chromosomes separating in meiosis 1 or failure of sister chromatids from separating during meiosis 2. So let's see how non-disjunction occurs during meiosis 1. Here's a cell in metaphase 1 and is getting ready to divide into anaphase 1. Normally, we would expect the homologous chromosome pairs to separate to opposite sides of the cell, but instead, a separation doesn't happen with chromosome 1. Instead, both chromosomes pairs are pulled to the same side. Because of this, you'll result in having 4 gametes with either an extra chromosome or missing a chromosome altogether. In the top gametes, they both receive 2 copies of chromosome 1 instead of 1 copy, and they retained a single copy of chromosome 2. The bottom gametes do not receive any copies of chromosome 1 since both went to the top 2 gametes. However, they did retain a single copy of chromosome 2. Non-disjunction can also happen during meiosis 2, specifically during anaphase 2. So while meiosis 1 occurred normally dividing the homologous pairs, in meiosis 2, the sister chromatids fail to separate on the chromosome pair number 1. And as a result, one of the gametes will have 2 copies of chromosome 1 and 1 copy of chromosome 2. Another gamete will have no copies of chromosome 1 and 1 copy of chromosome 2. And finally, the other 2 gametes will show up normal with 1 copy of chromosome 1 and 1 chromosome 2. Another issue again with chromosomal structure is chromosomal inversion. So in this case, a portion of the chromosome becomes detached and inverted before being reattached. While the effects on the organism are mild compared to the disorders, this new arrangement could lead to either an underproduction or overproduction of certain gene products due to abnormal gene expression. So each of those letters there represents a gene on that particular chromosome. You'll notice on top, D and C are completely inverted by 180 degrees, and at the bottom, F and E have been completely inverted. And finally, a translocation occurs when a segment from one chromosome detaches and reattaches to another non-homologous chromosome. Now, sometimes these have no effect on genetics, such as this reciprocal translocation, resulting in chromosome segments simply being traded. However, if a translocation occurs where a segment is permanently deleted or elongated without reciprocation, it can lead to deadly results.
So now that we have seen the reproduction of cells and the source of genetic diversity, how do particular segments of DNA, called genes, become inherited from parent to offspring? Well, in our next set of videos, you will learn about the patterns of inheritance and the basics of genetics.